Let's take a look at the chemical foundation of biology and of life. Have your book open as you watch this video. The material is followed very closely. This chapter is written extremely clearly, so I'll often just be sharing my favorite parts. Atoms are the building blocks of molecules found in the universe and also of all the cells and living organisms. In this model of an organic molecule, the atoms of carbon are shown in black, hydrogen in white, nitrogen are blue, oxygen is red, and sulfur is yellow, and they're all shown in the proportional atomic size. The silver rods indicate the chemical bonds. All biological processes follow the laws of physics and chemistry. So in order to understand how biological systems work, it's important to understand the underlying laws of physics and chemistry. This has become easier as models like this have become readily available. In this video, we're going to touch on these three topics. They don't seem like they run together terribly well, so I apologize in advance if the video tends towards the jumpy side, but it all wraps up into a nice package. So let's start with an understanding of what a single atom looks like. At its most fundamental level, life is made up of matter, and matter is any substance that occupies space and has mass. Elements are unique forms of matter with specific chemical and physical properties that cannot be broken down into smaller substances by ordinary chemical reactions. There are 118 elements, but only 92 of them occur naturally. At the most simple level, atoms are a single unit of matter. In the center of the atom, you find a nucleus. It has a positively charged protons and neutral charged neutrons. Surrounding the nucleus, there's a clouded field of moving electrons, each with a single unit of negative charge. In their natural state, the number of protons is equal to the number of electrons, so the overall charge of an atom is balanced. Neutron numbers can be variable. They're largely kind of like pieces of scaffold. Now, 99% of an atom is air. It's nothing, yet they make up everything that has ever existed or will ever exist. Scientists have organized elements into a periodic table, and each box of that table contains information about that particular atom. Here you see a single data box that contains numerous pieces of information to the atomic number up top, that's telling you the number of protons, and the mass number on the bottom, which is indicating the number of protons and neutrons put together. If you subtract the atomic number from the mass number, you can calculate the number of neutrons an atom has. The symbol in the middle tells you which element the information is referring to. Here you see carbon, but it has two different mass numbers. They're called isotopes of one another. They vary in their differ of neutrons, which alters the mass number. If you're curious, carbon-12 is the naturally existing form. As I mentioned, carbon-12 is naturally occurring. There are some other types of carbon that are made in the atmosphere as our Earth is washed in radioactivity. Carbon-14 is made from carbon-nitrogen consistently in our atmosphere. There's a regular ratio of carbon-14 to carbon-12 in every living thing as we breathe in new air and take in new sugars, and therefore new molecules of carbon-14. Once living things die, they stop taking in carbon-14, so their ratio is skewed. The carbon-14 and carbon-12 also start to fall apart at a very specific rate. Exactly 15% of the matter will revert back to its original form within 5,730 years. This calculation is known as the half-life of the carbon. The longer something is dead, the more off that ratio is, because it hasn't been taking in new carbon-14, and we can actually read that ratio and age a fossil. We call this process carbon dating. This is the periodic table. It was created by Dmitry Mendeleev in 1869, and it had blank spaces in it. He knew that some elements must exist, but they just hadn't been found yet. We've been filling in these spaces ever since. As you can see with the color coding, there's an incredible amount of information contained within the table itself. The elements towards the left have open orbitals, and those to the far right have full orbitals or full valence shells. Therefore, the elements that are in the row labeled 18, all the way to the right-hand side, are not reactive. Let's look at what those orbitals are and what they mean. We can judge the reactivity of elements by looking at orbitals and seeing exactly what they contain. The Bohr model of electron shells was developed by Niels Bohr in 1913. In this model, electrons exist within printable shells, labeled here as 1n, 2n, and 3n. An electron normally exists in the lowest energy shell available, and that's the one that's closest to the nucleus. Energy from a photon of light can bump it up to a higher energy shell, but the situation is unstable, so the electron quickly decays back to its ground state. In that process, a photon of light is released and energy is released into our environment. We'll get to the specifics of that strange sounding process when we talk about photosynthesis. Now back to those orbitals. The Bohr diagrams indicate how many electrons fill up each orbital shell. Group 18 elements, the one I mentioned before, have a full outer or valence shell. A full valence shell is the most stable electron configurations. Those elements very rarely react with anything. Elements in other groups have partially filled valence shells and will gain or lose electrons to achieve a stable electron configuration. This idea that outer shells need to be full is what leads to chemical bonding. We can fill in these shells simply by counting out electrons and placing them in their proper location. Let's look at lithium. You see it right there. It says Li. Lithium has 3 protons naturally and therefore 3 electrons. If I wanted to draw a Bohr model of lithium, I would use this simple base structure and I would write Li in the middle of my atom, add 2 electrons to the first orbital because that's all it's capable of holding. I have 1 electron left, so that 1 electron would go into the second orbital. If I was drawing maybe carbon that has 8 electrons, I would put 2 electrons in that first orbital, and now I have 6 left, so I would draw 6 electrons in my second orbital. If I had something that had a crazy high number like 18, I would put 2 electrons in the first orbital, and then 8 in the second orbital, and then whoever is left would go into the third, and on and on and on. It seems like an odd concept, but it's pretty easy to work out once you get used to it. We'll practice these. I mentioned that not having an outer shell that is full leads to chemical bonding, and there are actually 3 types of chemical bonds. We're going to look at all 3 and what the shells mean and what the bonds end up doing in the systems. There are 3 major types of chemical bonds: covalent bonds, ionic bonds, and hydrogen bonds. Let's take a look first at covalent bonds. Few atoms exist entirely on their own. They often bond with other elements in order to fill their outer orbitals. Here you see 2 hydrogens, each with 1 electron in the outer shell and therefore 1 opening. That shell can hold up to 2. You also see oxygen with 6 electrons in the outer shell and therefore 2 openings. Energetically, the least costly move for both the hydrogen and the oxygen to make their outer shells happy is to simply share that electron floating around the hydrogen. In essence, making 3 full and happy shells, and thankfully for us, also making water. That's what you see in this picture, and it's a great representation of a chemical reaction. Most chemical reactions are represented in a straight line like this, with an arrow in the middle telling you that there are reactants on the left, things that are going to do the reacting, and you see the product of that reaction off to the right. In some instances, more than 1 electron can be shared. Here you see that 2 electrons are shared between 2 oxygens. The bond made by sharing 2 electrons is stronger than the bond by sharing 1. So there's more energy here than in the previous example. The bonds created by the sharing of electrons are called covalent bonds. They're the strongest chemical bonds that we're going to discuss. Some covalent bonds are known as polar bonds. They have slightly positive and slightly negative ends. This is due to the largest atoms naturally having a little bit more pull on the electrons, kind of like a gravity idea. It makes the molecule that results from the bonds sit at a bit of a bent angle, like the one we see here in water. These polar covalent bonds act quite differently in aqueous solutions because they have an end that's just slightly negative and an end that's just slightly positive. Not all covalent bonds work in this way. Methane, for instance, is the opposite. It has a perfectly balanced charge, largely because of its geometric shape. Carbon dioxide is linear. It has a balanced charge as well. So we call those types of bonds nonpolar bonds. Not all bonds are created with this sharing fashion though. Here we see the outer valence shells of sodium, that's Na, and chlorine, that's the Cl. Sodium has one electron in its outer shell. It's far easier to give up one electron than to collect 7 more, so that's what sodium tries to do. Chlorine works in the opposite way. It needs to collect only a single electron. This is a helpful and convenient relationship, especially considering both are so naturally abundant. An electron will physically move from sodium onto chlorine, making sodium that has a full outer shell now, and chlorine that has a full outer shell now. Sodium would then be considered a cation, which is the name given to net positive ions. We say that it's net positive because that balancing act between the number of positive protons and negative neutrons has been thrown off. You lost a negative electron, so it's now positive 1 proton. The opposite is true for chlorine. It is now negative by a factor of 1, and it's referred to as an anion. This is because, remember, it gained 1 negative electron, so it has 1 more negative unit than it does a positive unit. These difference in charges in their ions actually make them bond together like magnets, and this is called ionic bonding. The third type of bonding is hydrogen bonding. It occurs most often and most strongly in water, and it gives water some very interesting properties. Hydrogen bonding occurs because of the polarity of water molecules. Let's look at what those tiny hydrogen bonds allows water to do. The coolest of chemical properties belongs to water. Water exhibits something called polarity because it has no net charge, but rather a slightly polar charge. Some molecules will actually move to get away from water because they have hydrophilic, which means water-loving, and hydrophobic, or water-hating, tendencies, and they condense to stay together. All of this condensation, the water-loving and the water-hating, is purely because of those slightly tiny charges. Being hydrophilic or hydrophobic is all about chemistry, and it's a property of water. Another cool property of water lies in the fact that it exists in 3 different states. Hydrogen bonding makes ice less dense than liquid water. It's because of the lattice structure of ice that it's less dense, and that allows it to float on top of water. Issues with global warming exist because if we melt all of the ice that's on Earth, like those sitting on land on the polar ice caps, the overall levels of the seas will rise, flooding hundreds of major cities and the homes of billions of people. As for the third state of water, a gas, we aren't used to a lot of that around here because it exists as humidity and fog in the air. 2 more properties of water are its high specific heat capacity and its heat of vaporization. When I say high heat capacity, what I'm referring to is the amount of heat that it takes to raise a gram of water by 1 degree Celsius. Water requires more heat than any other liquid to make this change, and that's actually the definition of the word calorie. It's how much energy it takes to heat water, 1 gram of water, by 1 degree Celsius. It takes water a long time to heat and a long time to cool. Hence, it's always gorgeous in California because the Pacific Ocean doesn't fluctuate very greatly. Winter's not long enough to freeze the water and summer's not cold enough to boil it off, so it helps to balance out the land temperatures that are changing more rapidly. The high heat of vaporization is kind of tied to that specific heat. It takes a lot to boil water and to move it from its liquid to gaseous state. This is actually really good for us. Water allows us as human beings to maintain our internal temperatures more regularly without expending a ton of energy. When you take a physiology class, there's a good chance you might learn about that. Water is also referred to as a solvent of life, and it's one of its great properties. It's a substance that's capable of dissolving other polar molecules and ionic compounds. The charges associated with these ionic molecules will form hydrogen bonds with the water, and then the water will surround the particle with individual little water molecule units. This surrounding is referred to as a sphere of hydration, or a hydration shell as seen here. When ionic compounds are added to water, the individual ions react with the polar regions of the water molecules, and their ionic bonds are disrupted. This disruption process is called dissociation. Dissociation occurs when atoms or group of atoms break off from molecules and form ions. Think about salt water. That's what's going on here. Another cool feature of water is surface tension. In cohesion, another property of water, water molecules are attached to each other because of hydrogen bonding, and keeping the molecules together at the liquid-gas interface causes surface tension. Cohesion allows for the development of surface tension, which is technically the capacity of a substance to withstand being ruptured when placed under tension or stress. This is why water forms droplets when placed on a dry surface rather than flattening out by gravity. Water also tends to stick to other things as well, and this property is known as adhesion. This can be seen in thin tubes. Thin tubes are called capillaries. Capillary action in glass tubes is caused by the adhesive forces exerted by the internal surface of the glass exceeding the cohesive forces between the water molecules themselves. That means that water has a tendency to be pulled more closely to glass than it is to the water itself. So water can travel up a glass tube due to the adhesive property, even though there's nothing sucking it up. Here's one of my favorite examples of surface tension: bugs that can seemingly walk on water. They're actually walking on the surface tension, but it looks really neat all the same. Another property of water, the final property we'll discuss, is its tendency to fall apart naturally in aqueous solutions in really tiny amounts. H2O will dissociate into lone hydrogen ions and molecules of hydroxide, or OH molecules, that carry a negative charge. Having large quantities of H plus ions is actually the definition of an acid. Solutions with large quantities of hydroxide ions are all basic or alkali in nature. The pH scale, or percentage of hydrogen scale, runs from 0 to 14. A pH of 0 is the strongest possible acid, and a pH of 14 is the strongest possible base. 7, where pure water would read on the pH scale, is considered completely neutral. This scale is read as a logarithm. If it's been a while since your last math class, that means that the levels increase by 10. So there are 10 times more H plus ions at a pH of 5 than there are at a pH of 6. And there are 100 times more H plus ions at a pH of 4 than a pH of 6. It works the other way too, a little bit backwards. There are 10 times more OH molecules at 11 than there are at 10. This diagram shows the body's buffering of blood pH levels. The blue arrows show the process of raising pH as more CO2 is made. The purple arrows indicate the reverse process, the lowering of pH as more bicarbonate is naturally created. Both of these processes happen constantly in our bodies. The changes in acidity in our bodies is actually what prompts you to breathe and exhale CO2 as it's created. The pH of the human body is generally safely kept between 7.35 and 7.45, meaning we're a little bit alkali or basic at all times. Now that you know some of the interesting properties of water, let's take a look at some of the properties of carbon, which is the backbone of life as we know it. Cells are made of macromolecules, which literally means large molecules, and they are put into 4 categories: proteins, nucleic acids, carbohydrates, and lipids. These macromolecules are considered a subset of organic molecules, and they're especially important for life. The fundamental component for all of these macromolecules is carbon. Carbon is the basis of life for various reasons, but one of its biggest assets is that it's capable of 4 bonds because it has 4 electrons in its valence shell and therefore 4 openings. We often use hydrocarbons as fuels, like propane for your gas grill or butane in a lighter. The many covalent bonds between the atoms in hydrocarbons are a great store of energy, and when you burn these molecules, that energy is released. Seen here is methane, which is a really common fuel. It has perfect geometric bonding, so this would be considered a non-polar covalent bonded molecule. Carbon is capable of bonding in numerous ways. The shape of the bonds can tell a biochemist a lot about its potential. When carbon bonding forms single bonds with other atoms, the shape is tetrahedral. When 2 carbon atoms form a double bond, the shape is planar, or flat. Single bonds, like those found in the ethane seen here, are able to rotate. Double bonds, like in the ethane, cannot rotate. So the atoms on either side are locked in place. Hydrocarbon chains can either be branched or unbranched, and we follow their shapes by connecting the carbons like playing connect the dots. Shape and structure is tied to function, which is the hallmark of the study of organic chemistry and the basis for a lot of cellular biology. Carbon can even form 5 or 6-membered rings, and they act quite differently than the linear chains we just looked at. Single or double bonds may connect the carbons in the ring, and maybe even a nitrogen might be substituted in. This might seem like a little vague of the purposes of a biology class, but for those of you who will continue on in science, you'll come to appreciate organic chemistry and all of its possibilities. Structure means so much in chemistry that even molecules that are made up of the exact same pieces behave quite differently when they're arranged in a different configuration. Think about houses. A lot of them are made of the exact same materials, but some can house 2 and some can house 10. Isomers are atoms that are made of the exact same materials, but they can house different bonds that serve different purposes. Molecules that have the same number and types of atoms, but are arranged differently, are called isomers. There are 3 major types of isomers. Structural isomers have different covalent bonding arrangements of the atoms, and that's what's seen here. Both of these chemicals will work quite differently, even though they have the same pieces. Geometric isomers have a different arrangement of atoms around a double bond. These two will act quite differently. Enantiomers are a special form of isomers that are mirror images of each other. It seems like these shouldn't function that differently, but stop for just a moment and take a look at your hands. Your hands are mirror images of one another, and they're definitely not superimposable and can't serve the same function. Enantiomers, again, are molecules that share the same chemical structure and chemical bonds, but they differ in their 3-dimensional placement of atoms, so they're mirror images of one another. Seen here is the amino acid alanine. The 2 structures are non-superimposable because there's a D form of alanine and an L form of alanine. In nature, the L forms of amino acids are used to make proteins. The D form of the amino acids are seen in cell walls of bacteria, but can never be part of their proteins. Similarly, the D form of glucose is the main product of photosynthesis, while the L form is a molecule rarely seen in nature. We can use our knowledge of organic chemistry to allow us to identify all manners of molecules, structures, and organisms. So again, if this seems a little bit weird for a biology class, I promise it has a purpose. Another hallmark for identification in chemistry and biology is the functional group. Functional groups are well-studied groups of elements that are often found together. They confer special properties on the biological macromolecules to which they are attached. This helps us to identify macromolecules. Only certain amino acids, for instance, are found on certain macromolecules. They participate in bonding and chemical reactions in a uniquely identifying manner. For instance, an OH bonded to the end of something makes it an alcohol. OH is therefore considered a functional group, and all alcohols act similarly. A carbon double bonded to an OH makes something a ketone, and that's considered a functional group, and it has a special range of characteristics as well. Hydrogen bonds between functional groups are important to the function of the macromolecules and may actually help them fold into and maintain proper appropriate shape for functioning. Hydrogen bonds are involved in various recognition processes such as DNA complementary base pairing and the binding of an enzyme to a substrate. That's what you see here. We'll talk about chemical bonding and why all of these hallmarks are important as we move through this semester. And on that note, we're going to end our video for today. If you need to better your understanding of this material, read the chapter and check Learner for any homework assignments.